Thanks, Jordy, for the introduction, and thanks to Fluidigm for organizing this great session. So I'm gonna start off with sort of a basic theme that's sort of the general overview of this entire meeting, which is sort of why do we want mass cytometry? What can this technology do for us? So hopefully this is pretty obvious to most people in the room at this point, if you're here, which is that mass cytometry can be thought of as analogous to flow cytometry, but by replacing these fluorescent markers with metal-based markers, you can now do more parameters, minimal spillover, low variation in your signals in the relevant mass range, low background. So all this comes together to allow you to really do a much greater breadth and depth of phenotypic and functional cytometric profiling than was ever possible before. So what this means is you can take a heterogeneous sample of cells and really dissect that heterogeneity on the basis of phenotypic and functional markers, and again, really deeply interrogate cellular function on a single cell level. So this is all very cool, and as Aaron's talk I hopefully made pretty clear, and many of our other talks will, you can do lots of really cool stuff with this data. So I'm actually going to take a much more basic approach for most of this talk, and instead focus on what are some of the initial steps that have to be taken to be able to actually establish a facility that is able to generate this sort of data. So very broadly speaking, you need a program that has an instrument. You need a CyTOF to be able to do this, which is also supported by an appropriate cohort of reagents. You need appropriate projects to take advantage of that instrument and your reagents. And you need a way of analyzing all the data that you're getting out of all this. So if you're like me, you spend a lot of time looking at spade trees. So I thought I would, to consider some of the heterogeneity within each of these categories, I'd try and display it in a more familiar way. So considering the instrument first, you have the CyTOF, of course, which is sort of the center of the whole program. But the CyTOF itself is a very sensitive device, and its performance and the data you get from it is highly related to a really strong program of maintenance and tuning and quality control. And in order to do this, you also have to have parts and consumables to support that element of things, and a good relationship with tech support at DVS, because you need them. So also the CyTOF, unlike, let's say, traditional flow cytometer, isn't entirely just a standalone instrument. You actually do have to have connections to ventilation systems in the room, and that's sort of an underappreciated and very important aspect of setting up a facility. So a good relationship, again, with facilities support and building maintenance is another really important factor when you're thinking about setting up one of these programs. So this is all well and good, but of course, in order to actually get this machine to do anything, you need reagents, which again can be divided into a few categories. So the sort of go-to cadre of reagents at this point are commercial conjugate antibodies available from Fluidigm directly. There is also conjugation kits, which allow you to conjugate your own antibodies, and which all require validation and titration. And then while antibodies are sort of the predominant theme of reagents that get used, there are also non-antibody-based custom reagents, ranging from fairly straightforward things like regular DNA intercalators to identify cells, or viability reagents, and also sort of fancier reagents like metabolic labeling reagents to be able to look at proliferating cells, and a whole host of new things that are in development. So putting these two together, you can now develop projects to take advantage of it. You need appropriately designed experiments, which take advantage of panels that are designed based on these reagents. These can be used to prepare samples, and then independent of sort of the standard project workflow pipeline that's going into the machine, you also have to have a fairly robust program of troubleshooting and assay optimization sort of along the way to support the standard experimental programs. So all this goes together to allow you to acquire some data. And an important aspect of CyTOF data acquisition is actually data normalization. So this is an important step to sort of account for differences in the machine over time and across institutions, and it's a way of developing much more robust data. And finally, of course, there's the visualization and analysis of all of this. So these are sort of all the components that one needs to think about when starting a new CyTOF facility. And so the next major question is, what should the role of the core, or the shared resource, be in setting something like this up? And there's a few different sort of models I'm going to propose for doing this. So the first off is sort of a focus really of the core on the instrumentation. It's kind of historically how a lot of new instruments are established in facilities. So typically if we buy a cell sorter or a flow cytometer, this has historically been the model we use, where the responsibility of the core is really maintaining the instrumentation and allowing users to generate data on the basis of this instrumentation. So again, in this case, in the case of CyTOF, this isn't trivial because in addition to acquiring the machine, as I mentioned briefly before, it does require pretty robust protocol maintenance. So it requires daily optimization, which has to be done rigorously with careful quality control considerations to ensure the data you're getting out of the machine is optimal. There's daily cleaning of the nebulizers and the lines. There's weekly cleaning of other glassware. Every part of this machine is made of a tiny, fragile piece of glass, so you need to have good hands for the whole process. And then you also need to have a really good repository of parts and consumables, because there are regular bits that need to be tweaked and replaced. And in order to have a good sample workflow or project workflow, you need to have everything on hand to be able to regularly keep things running well, including argon, which is another. So the machine essentially feeds on argon, and having an argon tank run out in the middle of an experiment is the worst. So it's really getting a good sense of how your inventory changes over time and how to keep everything in place to allow things to move along. So this is not trivial. And I think this is probably the minimal way in which a CyTOF can be implemented in an institution as a shared resource. The issue here is that investigators themselves are responsible for all other aspects of the workflow. So they're responsible for essentially developing, either purchasing their reagents from Fluidigm, conjugating and validating their own antibodies, developing other reagents, and developing their own experiments and collecting and analyzing their data. And I think this model can work in a specific sort of circumstance. And it's really when you have a core user or a core group of users who are already very familiar with technology or highly committed to developing technology. So for example, if someone from Gary Nolan's lab goes and starts their own lab, they'd probably be well set up to take advantage of a situation like this. Now here at Sinai, as sort of a research hospital and a clinical center, that was not really the case. We had a lot of investigators for whom it was pretty clear that CyTOF would be very valuable, but many of them were unfamiliar with the technology as a whole, and many of them didn't really have the resources to commit to sort of embrace and optimize a brand new technology. So our goal in establishing the CyTOF here, I think naively we initially thought we might be able to get away with sort of this sort of approach, and I think it became apparent very quickly we wouldn't. And so we sort of swung the pendulum all the way in the other direction and tried to make it essentially a completely full-service facility. And the goal here essentially being to facilitate access to every investigator at Sinai who might have any remote interest in using the instrumentation. And so so of course there's the instrument maintenance part, which I mentioned before. But another really key element to this was having a very strong repository of reagents available for investigators here. And this was formed in a couple of different ways. One was that we had an in-house repository largely derived from commercial antibodies sold by DVS. And the primary motivation for doing this is actually just cost savings, because an average antibody from DVS costs about $350 a pop for a vial that you can do 100 experiments with. But when someone wants to do a pilot experiment, they don't necessarily want to do 100 tests. And so let's say you just want to run 2 samples, but you want to look at 30 different parameters. You're looking at $10,000 worth of antibody you have to buy to do that pilot experiment. So that's obviously a very significant barrier to someone in being able to start an experiment. So our strategy was that we actually developed our own in-house repositories of many of the antibodies we thought would be useful and could then allow investigators to use them on a per test basis. So now by essentially paying $3.50 or $4 for one use of that antibody, you're only spending $250 on that experiment instead of $10,000. So this was sort of a first really important step to be able to facilitate access. And we sort of put a lot of thought into how best to develop that repository. And we sort of used a similar framework to what DVS does in designing their own panels. So we designed sort of a comprehensive, broad phenotyping backbone for both mouse and human that covers all the general cell types most people would be interested in. And then we developed sort of more focused modules to look at, let's say, T-cell subsets or B-cell subsets or myeloid subsets, depending on individual investigators' anticipated needs. And a key element of this was, we started off small with things that were admittedly largely familiar to me because I'm an immunologist, so it was a very immunology-focused development. But as time went on, we also supplemented this with a large number of custom antibodies, which were much more sort of on a per-request basis for individual investigators' projects. And rather than have those antibodies be specific to one investigator's project, we kind of took all of them and added them to the central pool to kind of continually grow the repository. And I really feel that the utility of the resource is almost directly linked to the size and validity of the reagents available. So there was also a very heavy involvement at the experimental design phase. So while the experiments themselves were generally conceived by investigators, there was a lot of education involved early on in the process. And a lot of it's just about appropriately managing expectations. One of the senses I got early on was that people were very aware of the pros of the CyTOF. It's a flow cytometer, but you can do 300 parameters. That actually isn't what you can do with a CyTOF. So a lot of it is sort of limiting, designing experiments to actually take advantage of technology, and also keeping in mind some of the limitations. For example, sample throughput is quite slow. So yes, you can look at a lot of parameters, but if you want to look at an ultra-rare population, 0.75% isn't bad. If you want to look at something that's 0.001%, as some of our samples do have, this isn't the right technology for you, because you don't want to have to spend 6 hours acquiring enough events to be able to see your cells. Similarly, if you work with really sticky tissues that routinely clog a flow cytometer, this is probably not the best platform for you there either. So it was really working with investigators to try and tailor their experiments to best take advantage of the pros of the instrument while minimizing limitations from the cons. Antibody panel design, again, those of you who have done some mass cytometry, is actually very, very straightforward in the CyTOF. It's much easier than flow cytometry. But nevertheless, it was something that was a little bit foreign to a lot of investigators. So rather than have them have to concern themselves with that, we took care of most of the antibody design as well. And likewise with sample preparation, which is, again, largely similar to flow cytometry, but we just figured it was another area that we could streamline everything. We also took care of most of the troubleshooting and acquired the data, normalized it, and also assisted with data analysis and visualization. So sort of in this model, what we were effectively doing was saying, let's discuss a project, you give us cells, I'll give you a completed figure for a grant or paper. So this was pretty nice. So it worked out really well for getting a lot of access early, getting a lot of investigators excited about the technology. But obviously, it was quite resource intensive on the part of the core, and it's probably not really tenable as a long-term option. And I think we're probably going to continue to offer the service in this sort of full service mode, but probably start charging a premium for it, just to account for all the time and effort it takes, and try and instead transition the core more to this sort of shared model that sort of divides the work up between both parties. And the key here is to take all the parts that are fairly straightforward and fairly simple and kind of start putting those more in the space of the investigators. So allow investigators to conjugate their own antibodies and take care of antibody validation and titration, but we still want to use this idea of having this central repository which you want to grow over time. And so, for example, if an investigator conjugates their own antibody, by contributing the antibody to the core repository, they can get a credit, which can then be used to buy a whole bunch of other antibodies. So it's a way that everybody kind of benefits. Similarly, experimental design can largely move to the sphere of the investigator, and we can continue to assist with troubleshooting. And I think for the most part, we're probably going to continue to acquire samples ourselves in-house at the core, but we may again move towards a model of training select users who are potentially very heavy users of the facility to start acquiring their own data. And the advantage of this approach is that it now frees up our resources to work on the things that investigators probably don't want to work on to the same extent. For example, really optimizing a lot of assays for general use and developing custom reagents through collaborations with chemistry, for example, and developing new data visualization methods through collaborations with bioinformatics, and really kind of keeping current with the field. So that's sort of the evolution of how things have moved over time. And I just wanted to briefly mention some of the key milestones right at the start that are important if you're thinking of getting a CyTOF and starting a new program. So one of the points I mentioned before is that you actually do need to have pretty strong support from your institutional engineering facilities in setting up an appropriate space for this instrument. And that should really get done before the instrument gets here. A consistent theme that I found, I was talking with Aaron and many other people, is that the CyTOF arrives and all of a sudden you realize there's 3 things wrong with the room that prevent you from being able to install it. So that can be nipped in the bud by, again, having a clear working relationship with engineering beforehand. There is a ton to learn about the CyTOF. It's an interesting instrument, and it's really at the interface of all sorts of technologies. So before it gets here, the person responsible for it has a lot to read. So at least in my case, in addition to reading every paper that had the word CyTOF in it, it was also going through a lot of old inorganic chemistry that I hadn't looked at for a long time, and physics, and being able to sort of get all these parts in place so that when the instrument actually arrives, you can hit the ground running. So DVS provides the installation and initial training. And the key after that is really becoming very familiar with the instrument. So this is just doing a lot of maintenance on it, tuning it, taking it apart, putting it together, figuring out how, if you loosen this thing, how it's going to affect your signal, and if you break this part, whether it's still going to be usable. And so really it's just becoming really familiar, and through robust testing, characterization, and optimization of your instrument. So moving on then to some actual biological validation. Initial testing I really recommend doing with DVS's panel kits. So these are sort of their pre-optimized sets of antibodies you can buy. That's kind of a nice, it's sort of training wheels for the CyTOF. And it's a nice way of getting started with things. And then moving on to getting a robust protocol for conjugating and validating your own antibodies in-house. And really these, and also troubleshooting and optimizing the protocols associated with using these reagents. And a lot of this is actually fairly fundamental. I mean, I'd done flow cytometry for 10 years, and it still took me a while to realize that fixation conditions for the CyTOF are quite different. And if you don't fix your cells properly, your data is garbage. So there's a lot of small things that need some tweaking as you're at this phase. And a lot of this, I think, is best accomplished through initial pilot projects with investigators. So here, these are individuals who don't necessarily expect that every experiment is going to work off the bat, and are willing to contribute the resources to really allow you to optimize a lot of these workflows. And in the context of doing these projects, you sort of familiarize yourself with all of these various aspects, and also end up building the repository that ends up being broadly useful to the community as a whole. And in developing some of these protocols, it's a good idea to sort of start slow with things that are fairly simple before you try and do what Aaron does, just because he makes it look easy, but it isn't. So I'm going to talk about one specific project, just as an example of some of this sort of pilot project work, to give you a sense of how some of this stuff evolves over time, and some of the troubleshooting and thought that goes into it. So this was a project with Cecilia Barron, who's an allergist and is interested in pediatric allergy. And the specific question she was interested in for the purpose of this talk was in peanut allergic children, when they get exposed to peanut, what are the early signaling effects that happen in those patients? Essentially, what's the early immune response to peanut allergy look like? And so the ideal strategy was really to use whole blood that was essentially minimally manipulated. We want to take blood, throw some peanut on there, and then fix it as quickly as possible to minimize any sort of environmental artifacts on that sample. And then use a comprehensive CyTOF panel to delineate a number of relevant cell types, and then overlay potential dynamic changes in activation of those cells in response to peanut. So we started off, as I suggested, by using the DVS17 parameter panel kit. So this is a nice, fairly comprehensive kit. And I'd already done some validation of this with PBMCs. But for the purpose of her studies, we needed to, again, confirm whether the antibodies that work in PBMCs also work in the context of whole blood. And also whether they work in the context of whole blood after it's been lysed and fixed. Because again, the idea here was we wanted to fix that sample as soon as we could post-stimulation to capture that dynamic activation status. So I'm not going to show you any data on this particular part, but suffice to say, many of the antibodies work pretty well. There's obviously some differences in whole blood. One is that a majority of the population is actually taken up by granulocytes, which are largely absent in the PBMCs. And consequently, some antibodies have to be retitrated. For example, CD16, which in PBMCs is largely going to be expressed on things like monocytes and NK cells. In the case of whole blood, is almost entirely bound up by neutrophils. So you need to throw more antibody in there to compensate. And there are a few other antibodies, like CD27, for example, that don't stain particularly well post-lyse fix. But after these initial optimizations with the standard panel, we then wanted to take this panel and sort of customize it a little bit to add in some additional markers of particular relevance to allergy. So for example, a lot of the Fc gamma receptors, the Fc epsilon receptors. We also wanted a means of identifying eosinophils, which we tested a few antibodies and ultimately settled on CCR3. And we also wanted to add in CD56 to identify NK cells, which were not in the initial panel. And so in each of these antibodies, we either had to acquire them if they were commercially available, which in the case of most of these, they were not. And in the case of these, we had to conjugate them, validate them, titrate them, confirm that they all work in the context we want to use them in. And there was some interesting troubleshooting that came about, which I'm going to touch on briefly. So here's just one example, and I think this is sort of interesting and a little bit weird. So this is a very commonly used clone for CD56, NCAM 16.2. So you'll notice that DVS sells 3 different CD56 clones, and in talking to people, everyone said this is the best clone that works really well. And sure enough, in PBMCs, it worked very well. You can see that you have a nicely defined NK cell population here. It's CD56 positive, CD16 positive, everything's great. But when we tried using this in whole blood, it performed very poorly. And it actually performed poorly in a weird way that I'd never seen before. So in staining whole blood prior to fixation, firstly, the antibody just didn't work well. You can see that you don't really, so this is, again, the whole blood population. This here represents your sort of leukocytes, and this is your granulocytes that are CD66B, so primarily neutrophils and some eosinophils. So you would expect all the NK cells to be in this population here. And you can see that you don't have particularly good resolution of the population prior to fixation. There's a lot of background staining here also in the granulocytes. And after you fix the sample, this thing just goes completely crazy. The neutrophils are all entirely positive. Every granulocyte population lights up. These guys here are actually basophils. And I mean, these are, I guess, the NK cells are actually contained within this, but there's a whole bunch of other junk in there. So this antibody clearly wasn't going to work in this application. So it turned out that we, through conversations with a graduate student at the Reagents Institute, we recommended trying this clone B159, which is actually a BD clone that they use in a lot of their phosphoflow experiments. And so we purchased this and conjugated in-house to 173 ytterbium. And you can see that the two clones actually perform quite similarly in PBMCs. The titration curves are a little bit different, but essentially you have the same population being stained. But now this also works very well in whole blood. You can see that the granulocytes no longer have this weird background staining, and then the NK cells are clearly defined, and they can be defined both pre and post staining. So this is now a clone that can be used in the context of this specific study. So another weird thing that we came across in these experiments, and I'm curious how many of you who work with whole blood have seen this before, but the first few experiments we found, the data looked really weird, and there was this kind of streaky population that was present across the samples, and it was just really messy. And for a while initially, I was like, oh, I guess this is just some sort of staining artifact or a problem with the instrument, and I tried fixing it with tuning. But ultimately, through doing some more tests, it became clear that these are actually eosinophils. And it turns out that eosinophils non-specifically bind to all antibodies if you try and stain them post-fixation and post-permeabilization. So to deal with this issue, we actually ended up doing some experiments and developing a buffer to help avoid this problem. So here you can see that this is a whole blood sample stained with CCR3 and CD66B. You can readily identify the eosinophil population. And now if you fix this, or if you take the sample and then also stain it with antibodies against CD3 and CD8, so T-cell markers, if you stain prior to fixing and perming the sample, as you would expect, the eosinophils don't express CD3 and CD8, which they shouldn't. If you stain them post-fix perm, you can see that they are now sort of non-specifically binding to all sorts of stuff. And so part of this project involved developing this blocking method that allows you to revert these back to the phenotype that you expect them to. So even after doing all this, however, a second issue now is that when we eliminate, it turns out that this might make you think that eosinophils are actually expressing CCR3 in these samples. It turns out this is all just weird background binding, and when you actually fix them, you lose all CCR3 expression. So it's still kind of hard to find them, but now because they're not autofluorescent or not auto, whatever the term would be, they're non-specifically binding to everything, they can be identified as being CD66 positive granulocytes that are negative for CD16. So at least we have a way of identifying them in the panel. So with all this out of the way, we now have a surface panel to identify all the cell types that we're interested in. And we now wanted to supplement this surface panel with some relevant markers to actually look at the activation status of the cell. And so the first aspect here was to take a standard basophil activation assay that's commonly used by flow cytometry and adapt it for CyTOF. And so this is based on, it's a degranulation assay that's based on expression of CD63 at the surface of basophils following activation. And so this just involved conjugation of the CD63 antibody and titration and use in the context of one of these activation studies. And you can see it works quite well. Unstimulated eosinophils express some CD63, and there's a dramatic upregulation post-activation. And then in addition, we wanted to supplement this with a panel to look at some phosphomarkers that were potentially of relevance in the context of allergy signaling. So with all these parts in place, we were now ready to actually put everything together and do the initial experiment that we hoped to do. So this was just taking 1 ml of blood from an allergic patient and stimulating it or leaving it unstimulated for 15 minutes with peanut, and then fixing it, staining it with a surface panel, and looking at some signaling markers. So first off, you can see that we were able to, the surface panel was able to allow us to delineate all of the major subsets that would be expected to be found in a blood sample. And then we could take this and look at the dynamic changes in the expression of specific markers in response to stimulation. So here I'm showing you the full change of CD63 pre and post peanut stimulation. And you can see very cleanly that the CD63 is highly expressed on basophils, and it's very, very specific. So you can see that this is unstimulated, and this is a stimulated sample. So this is cool. It was a nice proof of principle. But of course, if this was all we wanted to look at, why do a CyTOF experiment? This isn't very interesting when most of your tree is uninteresting. But the power of the assay is now we can also look at signaling. And so while CD63 may be a very specific marker of basophils, and if this is what you're using, you may think they're the only relevant cell type in the context of allergy. But now you can see that while P38 signaling, for example, is induced in basophils, there's also induction in some of the monocyte populations and in some DCs and PDCs. And looking at phospho-ERK activation, you can actually see that there's robust activation in PDCs in addition, increasing the potential populations involved in the allergic response. So the next step now is, again, figuring out what is the consequence of this activation in these cells using, for example, downstream functional readouts. And the cool thing about this sort of high-dimensional analysis with the CyTOF is sometimes, just by virtue of the fact that you've thrown so many things into a panel, you sometimes find things you weren't expecting. So a cool outcome that came out of this is that I had CD61 in this panel as a marker for platelets, which in an unstimulated sample are CD61 positive and are the only CD61 positive cells in the sample. But it looks like, post-stimulation, you actually have very robust, presumably aggregation of platelets with basophils following activation. It's a very, it's a highly specific activation. And so how much of this is a consequence of the subsequent functional aspects in the basophils is a topic to be investigated further. So that's just an example of some of the kinds of things that go into developing some of these experiments. And the next step that we're doing with Cecilia's experiments is to try and improve sample throughput and manage costs better by taking all of this and incorporating a barcoding strategy so we can multiplex samples and do larger patient cohorts. But I don't think I have that much time to go into that aspect of things. So I'm just going to end with a couple of final remarks, which is that I think hopefully what I've made clear from this talk is that if you're thinking about acquiring a CyTOF, more so than thinking of acquiring a CyTOF, you should really be thinking about establishing a mass cytometry program, because the instrument by itself is not really sufficient to allow you to do what you want to do. And so the investment in the instrumentation really needs to be matched by an investment in personnel and education of those personnel and of the community in general, and an active R&D program to really develop technology. This is focused on developing novel reagents, optimizing assays, developing new methods for data visualization and analysis, because I think what's really cool about mass cytometry is that it's already a powerful tool. You already can do really cool things with it, but it's still not very mature. There's a lot of room for growth. And I think really pushing the envelope in that regards is where the future is. And setting up sort of this second component here is what really allows you to stay current and continue to develop these aspects. So with that, I'm going to end to acknowledge sort of the overall program, sort of my vision of how I see the CyTOF fitting in at Sinai. So firstly, it's a core instrument, so I'd like to acknowledge the Mount Sinai's Dean's core program. We initially established it in the flow cytometry core with Jordi Ochando as the director, and recently we've transitioned it to the human immune monitoring core with Miriam Murad as the director. Projects, of course, come from all across the institution, but a lot of our key users are the Tisch Cancer Institute and the Immunology Institute. Cecilia Barron is part of both. And in terms of sort of future collaborations, I'm really hoping that close relationships with chemists, for example, in translational molecular imaging, are going to assist with developing new reagents, and collaborations with genomics and multiscale biology are really going to help with visualizing the data and developing new means of progressing in that area. All right, thank you. And her.
Adeeb. Hey. Is this on? I'll just shout. If you're getting all that data from 1 mil, it doesn't seem like sample loss is a problem, but as you know, for us, it seems like sample loss is a problem. Yep. It hasn't been mentioned yet. So can you comment on that?
So that is another limitation of the instrument in that it's not as efficient as a regular cytometer. So when you take 1 million cells and you put them in a cytometer, you're going to get pretty close to 1 million data points out of that cytometer. With the CyTOF, that's definitely not the case. You're only sampling about 20 to 30% of the sample itself. So I think one of the challenges in your experiments was that the initial starting sample was actually fairly scarce. There weren't many cells to begin with. And I find that the rarer your starting sample, the more dramatic your cell losses are throughout the process. So it turns out a mil of blood has a lot of cells in it. You may not be interested in a lot of those cells, but the fact that there's a big bolus of neutrophils in there really helps to, I think, stabilize a lot of the sample. And a lot of it, I think, happens at the level of sample prep. So that's sort of the assay optimization in the early phases. But it's definitely an issue with the instrument that we're sort of improving over time.
When it comes to running the identical samples or samples from a large patient cohort over a number of days or over some time period, have you identified any keys to limiting day-to-day variability or batch effects in the data?
So conveniently, the CyTOF itself has pretty good ways of normalizing the data. So there's these, essentially all the samples are run by spiking in these bead standards, and then the data is normalized to the known bead signal levels post-acquisition. So that sort of takes care of variations in the instrument. But I find that a lot of batch effects are actually from sample prep. I mean, if you're taking a sample and staining it with 30 antibodies, that's 30 potential sources of error where your hand might shake and your pipetting may be off. So I think in terms of doing those kinds of studies, the best way of minimizing batch effects is really to try and bank them and then run them at one time point. And I think a really great way of approaching that is barcoding. So essentially barcoding all your individual samples, pulling them into a single tube, and then processing them as one sample. I think, Michelle, I assume you're going to talk about barcoding a little bit in your talk?
No, it wasn't something that I was going to specifically address, but we do have a barcoding kit in development at Fluidigm, and we hope to be releasing it in Q2 of next year.
And in the interim, we're also, part of the chemistry collaborations we're working on are developing other ways of potentially barcoding samples to be able to improve throughput. And I think that really is a key advantage in that space.
You mentioned that there are difficulties with sticky cells or tissues. Do you have any experience with tissue samples, in particular cancer tissue?
Yeah, so far I've actually been pleasantly surprised with cancer tissues. I'd really expected them to make a mess of the instrument, and it's kind of been okay. The key is really good sample prep throughout the process. So most of the tissues we've used have been put through some sort of Percol gradient to kind of eliminate a lot of the epithelial cells and things like that, which tend to be problematic. We have actually run some human gut biopsies without any enrichment, and I think in that case the samples were rare enough that it wasn't enough of a problem. But that being said, we've had some investigators who are trying to run like heart tissue and things that have clogged the machine. So a lot of it is at the level of sample prep, and it's sort of something you have to determine empirically, I think, for a sample by sample basis. But it's certainly possible. It doesn't preclude the running of samples.
So Adeeb, as you point out, this is a great new technology, but it is still pretty clunky. So what do you or Aaron envision, or people from Fluidigm, in CyTOF 3, 4, or 5, so we can make this, in fact, even more useful and, in fact, more specific?
So I could answer that, but I think the better person to answer it would actually be Scott Tanner, who's speaking at the end of the day and is going to talk about the evolution of CyTOF technology, and I think is going to actually, rather than me hypothesize, I think he'll actually be able to tell us what CyTOF 3, 4, and 5 are going to be. So I'll leave that to him.
Scott, do you want to give a preview now or wait for the big impact at the end of the day?
More parameters, more cells,
Oh
faster,
yeah, more parameters,
cheaper.
better efficiency of cell utilization, higher sensitivity. All those things are what we want. When they're going to happen, I don't know.
Free cookies.
In my R&D group, we have 3, 4, and 5 in our heads. They're not in hardware yet. So, but we'll talk about a few of the changes. My talk is sort of going to say what are all the components, just like what Adeeb did. All the components. What is mass cytometry, and where can we today fix some of those things? Where tomorrow can we do it, and where next year can we do it?
I guess I will add that my sense is that the hardware is actually working pretty close to where it's going to be working, and I think a lot of the advances are really going to be in the reagent space and finding new ways to capitalize on the fact that we can label cells with metals, which is sort of a thing that people haven't really thought about to that extent yet. So like metabolic labeling of cells, by tagging actual pathways and things, I think is going to be the next major area of development. Aaron.
Do you find that users are mostly using pre-conjugated antibodies, or are you? Oh, thank you. Are users mostly using pre-conjugated antibodies, or are you working with investigators to come up with highly customized panels?
Yeah, so that's sort of been, I think at the early phase, there's been a lot more customization involved, and I think we're trying to move towards more sort of these standardized backbones. One of the limitations is, of course, the power of technologies, you can do different things with it, and so limiting yourself to standard reagents sort of limits what you can do. Of course, the advantage of our approach is that every custom reagent essentially becomes a standard reagent as part of our in-house repository, so that helps in that regard. But yeah, so far I would say that maybe 60 to 70% are standard, and the remaining sort of 30 to 40% are customized at this point.
So have you had any issues about the spillover over the channels?
So yes, a little bit. So that's another sort of managing expectations.
Mm-hmm.
I think there's a sense that CyTOF doesn't require compensation, which is true. It doesn't. That doesn't mean that there's no spillover. But it's very predictable. So unlike flow cytometry, where spillover is highly dependent on many different factors in the instrument, the two sources of spillover in the CyTOF, one is oxidation of your elements. So every element that oxidizes is going to produce some signal in the plus 16, the oxide form of that channel. So that's sort of one source of spillover. And another is isotopic impurities in the metals. So for example, if you're using 3 different gadolinium isotopes, you may see some contamination of one isotope because it's only 99% pure. But the nice thing is that they're very controllable and very consistent over time. So a lot of it you can get around by designing your panel appropriately so that you kind of know where it's going to be and how to avoid it. And I would say that as long as you're working in the space of about 25 parameters or so, you can easily design panels where spillover is essentially not a problem at all. I think it's when you start crossing that 30 parameter barrier that you have to put more thought into how best to design panels to take that into consideration. Reg.
You mentioned interface of translational imaging.
Mhm.
Can you expand on that a little bit?
Sure, it's early days yet, but essentially one of the things that became clear to me, or has become clear to me over the past few months, is that I can see all sorts of potential rooms for improvement in particular reagents or particular ways to make the instrument more useful, but it's way beyond my chemistry knowledge. So I've essentially been wandering around Sinai trying to find a chemist to help me out with some of these problems, and I think that's one of the limitations of us as a hospital is we don't have like a really strong basic chemistry department. And so it turns out that translational molecular imaging are just on the floor above, and they're a whole bunch of fun people. And so we've been chatting about trying out some new ideas for alternative ways of chelating metals and alternative ways of tagging cells, just to see if we can play around with some of that. So sort of rather than answering a specific biological question, it's more sort of technical development of reagents. Either way.
So you mentioned customers. Are you also looking to do contract research for industry? Have you started that? Are you going to go that way?
And we've had some requests, and I certainly think it's a possibility. At this point, we do have the bandwidth for it, and that our machine has free room on it to accept projects. So, and I think, Reg, you're probably a better person to answer that. I mean, I think Sinai's policy is that we will accept projects from outside investigators. It's just that internal investigators take priority. But as long as we have available room on the instrument, I think we're happy to.
So you named your xenophil fixing solution Secret Soup. Does that mean you're going to start a business selling that stuff?
That would be great. Given how much people sell buffers for, that would be awesome. But I think we're just going to publish it. So we'll see. Maybe we'll do both. Yeah, no, we'll see what the Mount Sinai Patent Office thinks about it. I don't know anything about money. Hence, I was a grad student for a long time. That's not a career decision you make if you want to get rich. All right, so I think we're going to take a coffee break for 15 minutes before we resume the session.